Welcome to Evolutionary Hologenomics, a monthly podcast from the Center for Evolutionary Hologenomics at Copenhagen University. I'm Anna Verissimo, your host for today. I'm a scientist and I also coordinate internal communications here at the center. If you are not a scientist or this is not your field, fear not, because in this podcast, we aim to break down all the complicated words and concepts while rambling about microbes and their interactions with their hosts, so that everyone can follow along and explore this topic together with us. The center is brand new. It started in January 2020, and it hasn't even had its official opening ceremony because, well, COVID. Nevertheless, it has 45 members across 9 research groups doing awesome science that will be discussed in each episode of this podcast. You can download it from your favorite platform or watch the episodes on the Center for Evolutionary Hologenomics' YouTube channel. Today is our very first episode and my guest is Aoife Leonard. Hi, Aoife. Welcome.
Hi, thanks for having me.
Welcome to the Evolutionary Hologenomics Podcast. Thank you so much for joining us. How are you doing on this fine, icy morning?
Cold. But I'm very good otherwise. Thanks.
Yes.
How are you?
We have coffee to keep us warm.
In your CEH mug. Amazing.
Yes. Don't mind if I do. Some
little product placement.
Exactly. Join the center and we will sort you out with a mug. This is how you know that you belong. So I see you also have your hot brew. So let's just get comfy and talk about hologenomics. And I'm excited as well too, because I've obviously did a little bit of digging and you are working on a very, very interesting project, which we will get to. But first, if you would like to introduce yourself, so let us know a little bit about you and what you do at the Center for Evolutionary Hologenomics.
Sure, I'm Aoife Leonard and I'm from Ireland, but I'm a postdoc here in Copenhagen at the centre. I started about 6 months ago on a project called the Earth Hologenome Initiative. It's an open science and interdisciplinary network of scientists who all are looking towards the same goal of standardized and reproducible hologenomic data sets of wild animals. Currently, we're mostly in Europe and North America and Australia, but we also have quite a few collaborators starting to build up in Asia, including Japan and Pakistan.
Okay, world domination is the plan then.
Exactly.
Knowledge, I guess, collaborative knowledge. So if you are in these locations or anywhere for that matter, and you do research with animals, then please do join the initiative. And by the way, is it everyone who can join this or just people doing specific tasks or jobs?
So anyone can join as long as you're trapping wild animals, which is the really exciting part of it. So we've got people all the way from like conservation managers to microbiologists, and then people even that aren't collecting animals actually and are more into the bioinformatics side of it joining the initiative. So there's so many ways to get involved. It's really exciting. But at the moment, we're really trying to reach an unbiased global sampling effort across the globe. So we're really interested in speaking to people who are sampling wild animals.
That's awesome. So I have 2 questions regarding that people listening also might have. Which is, well, first of all, can you tell us a little bit about the aims of this Earth Hologenome Initiative? So what kind of animals and samples you guys aiming to collect, and then what do you do with it? Sure. Just very briefly.
So like at the center, we're the Center of Evolutionary Hologenomics. So obviously with this initiative, we are focusing on hologenomic samples. And we're really lucky to be at the center at this and be launching this initiative right whenever we're able to actually really contribute to standardizing these sort of data sets. And we're focusing on wild animals from all over the world. Really, we've got people sampling from bats to marsupials to salamanders. And from that, we're trying to get paired genomic and metagenomic data from the samples so that we can have this hologenomic approach. And that means that we're looking for samples that are either anal swabs or cloacal swabs or feces, and then maybe also a tissue or blood sample from the animal. And that will allow us to characterize both their microbiome and their genome of an individual species.
Okay. Now, one of the purposes of this podcast is to kind of just have these awesome discussions because I'm very nosy and I love science and I love to see what people in the center are doing, but also to kind of educate not only the general public and on what is all this weird jargon that scientists use. So guys, don't go anywhere. We will explain everything and hopefully you will learn something today and be very excited about learning some science. But it's also very useful even for scientists who are not doing the same kind of biology work that we do, because nowadays all the research is multidisciplinary. So not even though everybody is a scientist, not everyone speaks the same language. So if you say hologenome, it might make some sense to some biologist or microbiologist or ecologist doing a specific type of research within ecology. But we also need bioinformaticians, mathematicians, and all these people who do mathematical modeling to analyze all this humongous amount of data that is being generated. And so it's super, super important that people actually speak the same language. So even between scientists, it's super important that we explain what all these weird concepts are. And since this project is called Earth Hologenome Initiative, I love the fact that it's on the name Hologenome, because our center is the Center for Evolutionary Hologenomics, and this podcast is called Evolutionary Hologenomics. Therefore, hologenomics and hologenome is something that is going to come up in every single episode, I reckon. So, Aoife, you are the first guest, so you get to do the honors. Would you tell us very briefly what is this hologenome thing?
Sure. I mean, we scientists love our jargon, and there's even so much debate on definitions within the field, let alone between other fields in an interdisciplinary manner. For me, personally, hologenomics and how we use it in the Earth Hologenome Initiative is the joint analysis whenever you look at the individual and their DNA, their genome, as well as all the microorganisms in their, like on their host, including their gut microbiome or their skin microbiome, or even their like oral, the bacteria in their oral cavity, all the DNA associated with those organisms combined into one analysis. Would you agree with that?
Yes, and that is one big analysis. That is a lot of data, and in a way, it's super nice to, instead of seeing microorganisms that live in our gut or on our skin or like anywhere in the body or on the body, and the host, us or animals or whichever is the subject, as separate, but actually as entities that live together and definitely interact and influence each other in many different ways. So yes, I would agree with that. So hologenome, in a way, is the combined DNA information, if you will, of all the microorganisms living in and on a certain organism, plus that organism's DNA as well. And then we can learn a lot from that. And then hologenomics, I guess, is just a field that looks after that. So we've got at least 2 new concepts down this in this podcast. So that's very, very good. And then I'm a little bit more curious because I have seen images of some very mysterious black boxes. So you mentioned that people who collaborate in this initiative can get some swab samples. And you guys, as far as I understand, you can send them a specific kit because you already mentioned that you want to standardize your methods, which means that if everyone uses the same kit, then everyone kind of does it in the same way. And then you get the same kind of samples done in the same way. And then you can analyze them further in the same way as well. So what is in this mysterious black box?
So you've really hit the nail on the head with the whole aim of the Earth Hologenome Initiative is just collecting a standardized global data set. And to do that, samples need to be preserved in the same way, they need to be collected in the same way, they need to be processed, and the samples need to be processed in the same way. And we're really lucky to be able to be at the start of this and nail it down from the get-go. We can actually send out these kits around the globe to anybody that's sampling wild animals, and they contain tubes with pre-filled preservation buffer and pre-labeled, as well as the swabs that you could use for swabbing the anal cloacal swab or the oral swab, or even skin, like I mentioned earlier. So you have everything you need to conduct standardized sampling in the wild. And even the kits even have these racks on the inside of the lid, so you have a base already for your sampling, where you can put the tube in and then unscrew the lid all with one hand, all while holding an animal and taking your swab. So they've 3D printed these racks. It's really awesome. We have collaborators from over 40 different countries around the globe so far, but obviously not everybody's able to sample right now because of COVID. But we're really lucky, and we've sent them all the way from different countries in Europe to North America, and we're starting to send them to Australia now as well. So it's really exciting.
That is so cool. It reminds me like of all these kits that you can get from the shop, like these science kits, except you are actually doing real science. It's not just like a practice at home kind of thing. So that is, with the convenience that you get this box and you go to the field, and then everything you need is just there, and using it and sampling whilst you hold the animal, and you use your free hand to do everything else. So somebody really had put a lot of thought into how to make this kit really, really straightforward and super easy to use. You said that you already have about 40 collaborators, and but you're looking for way more.
We actually have like 140 now. Sorry, I meant 40 different countries.
Oh, sorry. Yes.
Yeah, we are obviously like trying to get as many people on board as well, but we're really trying to make sure that we have a totally unbiased sampling effort across the globe as well.
Cool. So you have about 140 collaborators already, and your plan is world domination of data collection in a standardized way. So if other people who are sampling animals or capturing animals for some reason in the wild, if they want to join this initiative, how can they do that? So this is your time now to share like all the social media links and websites and addresses and like anything that people can either follow, even if they're not doing research, but they're just interested in knowing the outcome and following how the project is doing. But also, if people actually do want to receive these kits, what do they do and where do they go?
Sure. So the first thing is just get in contact with us and let us know what you're sampling. We'd love to hear what everybody's doing in the field and how everybody can get involved. There's so many different options, and how, especially even if you're not sampling microbiome stuff at the moment, if you're just capturing an animal for your own research that might have a completely different objective, while you have that animal, why not like maximize your research output and maximize, if you're capturing an animal, you want to get as most out of that capture as possible. So why not? And you can follow us on at eHologenome on Twitter if you want to find out more. And we love to share people stories of who's sampling for us at the moment. Also, if you are sampling with us at the moment, please send us your photos, because I love to see them and see these kits in action, because I'm inside all the time and I want to see people outside, please. So there's so many different ways you can get involved. Follow us there and just fire us an email at ehi@soon.ku.dk, and we can chat about what you're up to. It'd be great to hear. Get you a kit sent out.
Yes, that sounds good. So please do. And I will link and add all of these email addresses and website addresses and all the social media handles to make sure that you do get in contact with Aoife and share your pictures with us, not just with her, but with everyone, because we love to follow along and see how everyone comes together to generate lots of data. And just having a little browse through the website here at earthhologenome.org, I can see that there's a lot of really nice pictures and lots of information about the project. And one of the things I love here is that you state the ethical principles for the project. So collaboration, openness, and transparency. That is really, really nice to see that a lot of people now are moving towards this rather than competing against each other and then in the end having less data. And as you just mentioned, if you are already sampling an animal, why not just get the most out of that rather than having to go and capture and hassle another animal? But these, as far as I understand from this kit, these are totally non-invasive, right? So you have a swab, or if you get blood, still you don't need to kill the animal, you don't need to injure the animal in any way. You just capture them, and then you just basically get a little swab, right?
Exactly. So no, all samples have to be collected from live individuals. So that's another part of our standardized protocols. Just because the microbiome can change so quickly after death as well is a big part of it. But then they're captured from live animals so that we can also get the host animals' associated metadata, which is going to be an important part for our analysis. But in terms of our ethical principles, it's such a core part of the Earth Hologenome Initiative, because not only do we want everything to be standardized and comparable, we want it to be open access. So even the preservation buffer that's in the tubes, it's non-commercial, so that we're really hoping that once this is completely standardized, everybody else will be able to make this themselves in their own labs. And that will really help the longevity of the project as well, because if we're reliant on commercial kits, even for DNA extraction or for the preservation of the samples, that kit might just be discontinued soon. You never really know. And that's our standardized protocol out the window. So we're really working on making sure that everything is completely standardized, non-commercial, and also cheap, which is very key for a lot of labs.
Long life to the Earth Hologenome Initiative's kit, which, by the way, the box looks amazing. The branding looks really, really cool. It looks like the proper thing. So even though you keep saying it's DIY, which is in a way, it looks very professional as well, and very, very cool. And obviously very practical to take into the field, and then it's standardized, which is excellent.
Which is a big part of it. So we have these robots in the lab, which I'm obsessed with, because they will pre-fill the tubes for us and also label the tubes and print out the labels for all our boxes and kits. Yeah, it's called Bettina. I love her.
That is amazing. I used to work in 3D bioprinting, and we also named all of our robots. We had Rebecca and Regina. So I totally get it. So Bettina is your machine that fills up all the tubes properly, and then you label everything and you can scan everything. So everything, you know what you've shipped, where to, and then when you get something back, you know exactly what's happened with it, and then you can carry on processing your samples.
Yeah, we even have Opentron robots that our amazing research assistant Martina is trying to completely perfect, that will even be able to do RNA and DNA extractions for us. So she's getting there, and soon that will be done, which will be very exciting.
So she's like the trainer of these robots.
Yeah, exactly. She is the robot whisperer.
Very, very cool. And you also mentioned that you have all these different animal groups, so all these different species. Your aim, I guess, of this world domination is to have all the species, but I imagine that's a little bit hard. And also, if you have 500 species, but then only one data point for each one, you don't get as much information, and you can't do as many comparisons as if you actually have more samples per species or per related species or ecological niche or whatever it is that you're looking at. So are there any target species at the moment?
Yeah, we actually have quite a few. So like you said, we're trying to sample as much as we can, but there's a lot of patterns in what people sample. People tend to sample things that are easy or are important for for conservation issues. And so we noticed quite a few patterns of what people were collecting, and we're able to start our own projects within the initiative, which is really exciting. So one of the first projects to come out of the Earth Hologenome Initiative is squirrel hologenomics, where we're comparing or looking at, both the Eastern gray squirrel and the Eurasian red squirrel.
Okay, so the name of them basically hints that they come from different places in the world.
Yeah.
Do they live separately? Do they live together? And is that natural, or man had a little bit?
Of course, man had a handle in it, Anna. Of course. So, okay, well, we're sampling them. So for all the species, we try and get at least 10 individuals per species and then 10 populations across their global distribution, just as like a general rule of thumb. But then for our projects, we maybe try and fine scale that a little bit more whenever there's interesting questions involved. So for our squirrel hologenomics, we are sampling the gray squirrels in their native America, Northern America, and they're invasive in Europe, mostly in Britain and Ireland and Italy. So we're able to sample the gray squirrels in both continents, and then the red squirrels in the native Europe across their global distribution, all the way from Ireland, Scotland, Italy to Japan, including Norway, Germany. We're going hard here to get their global distribution down. So where they're also completely live by themselves and they haven't been impacted by gray squirrels yet, and also where they are declining because of the gray squirrel due to like a variety of issues, such as the virus that gray squirrels give them, as well as interspecific competition.
Right. So just to kind of get everyone on the same page on what an invasive species is, you're not saying that the gray squirrel came on a boat rowing all by themselves and then invaded, right? That's what humans do. So whenever a species in nature is invasive, what does that mean in terms of how does it get to somewhere? And then why is it bad? Or what does it make it an invasive species, and how is that bad for the local species?
Okay, so usually whenever a species arrives in a new area, the grey squirrels were managed to arrive, and because they have the same sort of ecological niche, which means that they have the same food, they live in the same sort of areas as the red squirrels, they're able to outcompete the red squirrels, which is a bit of an issue. So we're seeing a decline in the red squirrel population. And that happens with so many different species. And invasive species can also harm, not just by competing with other species in their ecological niche, but just damage to the native environment. There's so many ways invasive species can impact by so many different ways. Even the grey squirrels have a huge impact on forestry in Britain, costing millions of pounds a year. This is worth the damage. I actually did my PhD as well on insects that were crop pests. So they spread with the global food trade on whenever we were buying seeds across the world in North America. These little guys just hitched a ride and now cost millions of pounds worth of damage globally in crop destroying mechanisms.
Yeah, so unfortunately that's happening with a lot of species all over. And so right now, hopefully with these studies as well, you will be able to get some data on both species, especially where they coexist. But you also mentioned that you are studying red squirrels, for example, in their native ecological niches, but across different types of environments, right? From completely sort of isolated, living in the woods, away from people, to now a more urbanized setting as well. And this, I guess, is where they coexist more with the gray squirrel.
Yeah, I think it depends on the location. So gray squirrels are particularly adapted to urban environments. And so especially around the central belt in Scotland, gray squirrels live in isolation from red squirrels. And that is just obviously another thing that humans have managed to do, is just destroy the rural environment. So a big topic in ecology is how species are adapting to this urban environment And that's one of the questions we're going to address, looking at both the grey and the red squirrels. So not only are we sampling them in their native and invasive habitat, we're also sampling along a gradient of urbanization. And on top of that, we're also, by having this global distribution, we can look at how they adapt to different climates. So that's also why we're looking at the global distribution of the red squirrels in Europe.
And on top of that, because now this is the era, at least in our center, of the hologenome, this would be like one study that people would do, which is just looking at the squirrels and their behavior or their genetics or their whatever, but which is also very important data. But now the Earth Hologenome Initiative is going beyond that, and you're not only looking at behaviors and ecology and physiology and genetics of the squirrels, of the different species and of the same species living in different environments, but you're also looking at the microbiome, so all the microorganisms that live in them and on them, or just in them at the moment. What kind of samples are you getting for the squirrels?
So for squirrels, mostly we're just getting fecal or anal swabs. And that'll characterize their microbiome. But to make sure we have enough host DNA for their to characterize their genome, we're also hoping to get oral swabs or even like a little tissue clipping. And then from that, we will get enough DNA to properly characterize their whole genome. So exactly like you said, we are the Center for Evolutionary Hologenomics, obviously, and we are the Earth Hologenome Initiative. So in terms of all these questions that we're asking, it's how does their microbiome contribute to their ability to adapt to these novel environments, and how does the microbiome contribute to their ability to compete against other species? And I think looking at it from this way is what makes it novel.
Yeah, so this is a relatively new field or a new way of looking at things, where you have a lot more integration of many different things, and hence why now we also need to work not only with biologists and ecologists, but especially with very smart people who do all these complicated bioinformatics and mathematical models, so that we can integrate all this kind of data. So squirrels, you're getting both those kinds of samples. And then for other species, you mentioned that depending on the species, then you might want to swab from different areas in the body. Why is that? And what kind of different groups of animals would you sample from different places in the body?
Sure. So it depends on the species of what we think is the important aspect that might contribute to the questions we're looking for. So I'm an evolutionary biologist, and I'm interested mostly in local adaptation. And we know that for instance, amphibians, that their skin microbiome can really influence how they can adapt to new environments. So for an animal like an amphibian, we would really think like a skin swab would be particularly important. But so there's a slightly different protocol for there, where you also don't want to get too much environmental DNA in those swabs. But then that would be less important maybe for an animal like a squirrel. So we don't rely on skin swabs to characterize their hologenome.
Yep, that makes sense. And so just I think people know what amphibians are, but just in case, can you give us a few examples of different groups of amphibians?
Well, some groups that we're sampling at the moment include salamanders and frogs are the two big ones that people tend to be sampling, especially the alpine salamander, which is a personal favorite.
They are very, very cute. Yes, I grew up with salamanders as well. And then I guess toads and newts are also amphibians. So that's the sort of animals we refer to when we say amphibians. And so I can imagine that from swabbing all these different types of areas in the body, so if it's either in the mouth or the anus or skin or whatever, then you also get different percentage of genetic material, so DNA, from either host or the biome, so all the microorganisms living in or on. Do you have an idea of how much more or less you would get from different types of swabs in different parts of the body?
Well, for that reason, we're conducting quite a lot of pilot studies because we don't know how much host DNA we'll get, maybe from a fecal swab or from feces or from an anal swab. So then we need to make sure that we're going to get that, and also that our preservation buffer is doing its job, and also then that our extraction protocols are able to get that and are able to pick up that extra host DNA and not just the microbiome. So we're conducting quite a lot of pilot studies at the moment, and for squirrel whole genomics is one of them, because quite a lot of people can't get tissue samples because red squirrels are obviously endangered, and so that's an issue there. So we really would like to try and make sure that we can get enough host DNA just from their fecal or anal swabs. So I actually got to go to the zoo last week, which is very exciting, to try and get a rodent, because nobody in the department has a pet rodent, which is quite shocking, I thought, for a load of biologists, probably with loads of kids who want pets at home. So I went to the zoo last week to sample a guinea pig oral swab to try and, which then we're going to sequence and see if we can get enough host DNA out of it. And then that'll become our standardized protocol for squirrel whole genomics. And we know that then these non-invasive techniques will enable us to properly characterize both the genome and the metagenome without relying on tissue or blood or anything like that.
Yeah. Okay, so once you get your data back, the results, then you will know from this guinea pig what percentage of the swab that you got is guinea pig and what percentage is microbiome. But I think another important thing is that sometimes we don't think about is that everything we eat, or a lot of things we eat, actually have DNA as well, because plants and animals and all these animal products or plant products, all of this has DNA as well. So then how do you factor in that as well if you're doing, for example, well, I guess it doesn't matter if it's a oral swab or anal swab, somehow it's either end of something being eaten and processed and then thrown away. So how do you expect to see a lot of that, and in what kind of samples, and how do you get rid of that when you do your analysis?
Even when I was sampling the guinea pig swab, when I took it out, it was bright yellow. Like the guinea pig just had a cheek full of straw. So I would not be surprised if whenever we sequence this, it's 99% straw, to be honest. So we'll have to look into that further, maybe get a couple of squirrels, hopefully, or a few pilot samples from collaborators, and then we can actually do it properly.
Yeah, so again, a lot of it has to do with robust sampling methods, just to make sure that you do get enough for your analysis. And then once you get all this bunch of data, which is basically the combined DNA of everything that touched this swab, so the host, the microbiome, the food, and who knows, if you're not wearing gloves, maybe a little bit of you as well.
Yeah, if you didn't eat the swab, at the moment tea, but 90% of the time it would be just a whole load of chocolate, to be perfectly honest.
So not much need for complicated bioinformatics. It would be like 99% chocolate, 1% human.
I'm not sure you'd be able to properly characterize my genome from an oral swab.
Yes, very, very nice. So yeah, definitely we need robust everything in every step, from collecting the sample to then analysis methods and ways of getting as much as possible of the DNA or RNA or whatever your analysis is out. And then once you have this bunch of data, you need very talented bioinformaticians and robust methods to actually be able to separate all these things and then be able to discern and only look at the data that you're looking for, either for the host or the microbiome, or actually, depending on what your study is, maybe you are interested in knowing what they're eating, and then you will have the data there as well.
Exactly. So some people do use these types of samples to do dietary analysis. That's not the goal of ours right now, but it's definitely something that could be done Although I'm not sure you would need to have done that with the guinea pigs because it was very obvious what their diet was. You don't need sequencing for that. Yeah.
Yeah. So speaking of that, what kind of so you said you have 100 collaborators, and I guess, well, I don't guess, I know that in the Center for Evolutionary Hologenomics we look mostly at DNA, and now we're starting to look at RNA as well, mostly, and then some projects have collaborations at looking at proteins and metabolites and things like that, which is basically things that the cells make and then put out, and they go in the body and then they interact within your body and between you and your microbiome and so on. But so within the network of the Earth Hologenome Initiative, what kind of analysis and fields and questions do you have apart from DNA? Do you have somebody, for example, looking at behavior of these squirrels in the UK, for example?
So that's actually a good point in terms of why we don't just want people collecting samples and submitting them to us at the initiative. We want it to be as collaborative as possible because we're not doing behavioral stuff at the moment necessarily, and we're not doing metabolomics, but why not? So when we launched Squirrel Hill Genomics, we were actually talking to some researchers in Norway who are interested in the energy, I guess, consumption of the squirrels. So they have these boxes where they can measure the energy consumption from the squirrels, or their metabolism, sorry. So there's so many things we wouldn't have even thought about. And that's why it's so great to have so many people involved and have this really truly interdisciplinary network. Because as well, not even just like making the most of somebody collecting the odd sample and being like, oh, sure, I'll just get an extra swab for these guys over here. There's also like making the most of those samples as well. Like, yeah, you can characterize as metagenome, but why aren't we also looking at all these other questions associated with that? So it really is just also trying to be as efficient as possible with samples, because like you said, these are live animals and they can take a long time to collect these samples. So why not just try and be as efficient as possible?
Yeah, and it works out great, like in so many ways. So it's less hassle for the animals, because if you already have it in your hands and you can just from the same or just one extra swab, you can get so much more information. Then you don't have to chase them in the park or the woods every single day and stress them out, which is good. Then it saves resources as well, because obviously research needs money and every field trip costs money. And then if you can collect all this information and integrate it from the same individuals, then there's just so much more integrations and comparisons that you can do.
Yeah, exactly. There's so many things that we would never have thought of. And then also by combining the data that we might have collected for and what somebody else would have thought to have collected, it just opens up a world of new questions. So I think it's really exciting.
It is very, very cool. And so people can get in touch with you, as you already mentioned. And on your website, I see that there's actually two different types of interactions. So people can either join a project or they can come up with new projects.
Exactly. So you can join one of our existing projects like Squirrel Hill Genomics if you are currently or able to start sampling red or gray squirrels across our global distribution. But we do also have other projects coming up, such as a project using bats and also a project in Australia looking at marsupial evolution and patterns of convergence with their placental mammals in the same region, and compare them also to placentals in Europe, so we can understand how the microbiome contributes to this convergent evolution. So there's so many different ways you can get involved if you're collecting these types of samples, but also if you just, we don't know what projects are going to be out there in the future. Like I said, a lot of it just comes from patterns of what we see, what people are going to be interested in, and what people are collecting at the moment. So get in touch and it'll just be really exciting. Who knows?
Yeah. So just have a chat with Aoife and see where it goes. Definitely. Collaboration is a wonderful thing. So you just mentioned, because we want to go over all these words, because some people might know what it is, some people might not know what it is. So marsupials, in Australia, of course, because that's where most of them live. So basically they have this pouch, and then their newborn, who looks a bit like this little blob that is not quite finished, lives in there and is protected and gets the milk from the mom. And then you mentioned placentals, which I guess everyone knows placenta, but could you just clarify what placentals are?
Yeah, a placental mammal is just an animal which gives birth to live young after developing them in their placenta inside their womb.
We humans are placentals, right?
Mm-hmm.
So we cook a baby in the oven and we have our placenta that is like this very nice, comfy cushion full of blood vessels and whatnot. And then kangaroos, so our babies are ready to go once they come out. And the kangaroos, they also give birth, but then they have this pouch where the baby basically finishes developing because it's not quite ready. Yeah. But anyway, that's a very, very big difference, obviously. So when we talk about evolution, we don't have pouches and some animals do. So obviously, even if you're not a biologist, you can see that this is a big difference in terms of how species evolved. So that's a really nice study.
Yeah. So even then, sometimes between marsupials and placental species, they still adapt very similar morphologies of but because they have different evolutionary histories, so they evolved completely separately. Marsupials over here and placentals over here. Within that, they still evolved to look very similar. So and need all this have the same ecological niche, and that's what we'll be studying with convergent evolution.
Yeah, so thank you very much. That was another concept since you mentioned it. So convergent evolution is when two species evolve separately, but somehow towards the same either function or look or whatever it is. So they did not come from the same ancestor and then diverged. So that would be divergent. But they are actually separate, and then somehow because they needed something or whatever it was, then now they developed something very similar. So that, and again, you guys are going to try and see the involvement of the microbiome in this convergent evolution as well.
Exactly. That's a new postdoc, Raphael, will be taking and coordinating that project, which is going to be really exciting as well, to have more people on board the Earth Hologenome Initiative.
So basically, this is a very, very global initiative, which is super cool, with people from all over looking at many different species and many different microbiomes and interactions and environments, and with so many different scientific questions. That is very, very cool.
It's very ambitious.
Yes, it is. But in a way, and all of this, because again, it's called the Earth Hologenome Initiative, is to shine a light on the role of the hologenome in all of these processes and in answering all of these questions that people might have asked before, but instead of analyzing everything as a system with so many components, they perhaps just analyzed a very small fraction. So this is the novelty.
Exactly. I think like biology in general has been so focused on how genes interact with their environment to produce a phenotype, which is just how it produces an organism. But nobody's been looking at how their metagenome interacts with that environment, and also how it interacts with the genome of the host animal. So it's adding like another entire layer of complexity that has been largely ignored for quite a long time, or just looked at it separately and how it interacts with the environment. But it's this interaction that we're really focusing on.
So some new words. I'm always like on the lookout for new words. So hologenome, we already covered. Genome basically is the genes, very loosely, or DNA. And then you just mentioned metagenome as well. So what is the genome and the metagenome?
So the genome is the entire DNA of an organism, and the metagenome is all the DNA of all of the microorganisms within the host organism. combined. And together, those are the whole genome.
Yes,
so that is why we're here.
Indeed, we have a whole center dedicated to the study of the hologenome.
Product placement, love it.
Yes, I mean, join us, by the way. So you not only can you join the Earth Hologenome Initiative, but you can also join the center. And if you are interested and you join us, then you get a mug. This is how you know you belong. But also you get really, really amazing colleagues and so many people from different disciplines who are very much looking forward to collaborating with each other. And I think this podcast, although it is for everybody, it kind of came from this need that the people in the center felt to know what everybody else is doing so that they can see how they can collaborate within the center. And then, of course, the Earth Hologenome Initiative takes it beyond the center. And actually, you guys want everyone in the world to collaborate and build and analyze data together. And because this is open, so it's not like, give me your samples and then I will publish the paper. But actually, you are also looking into people having inputs and getting something out of this project. Because for the people listening or watching who don't know, bioinformatics nowadays, it takes time. It's not just you press a button in the computer and then you get your data. So definitely we need brains and hands on deck to do all this. And we need very smart people from all the different disciplines to pose questions and to integrate the data and analyze and interpret the data in the light of their own knowledge and skills, that by ourselves we don't have this combined knowledge, right?
Exactly. I mean, nobody has all of this knowledge all by themselves, and we really do want the initiative to be, the whole point of the initiative is to foster these sort of collaborations. And at the end of the day, after we've standardized all these protocols and everything is completely non-commercial, we want to be able to just roll that out to other labs that aren't able to maybe necessarily buy these commercial kits themselves. So it's really not just a case of send us your samples and we'll publish, thanks very much. We want people to be involved at every step along the way. And then all of these samples will be added to a biobank, which will be totally open access. Everybody will be able to know what is available. So just, you can get in touch whenever you want. That is totally fine. And also, hopefully then people will be able to carry on the initiative and join the initiative from their own labs, which is really the end goal of the whole thing and what open science is about.
That is very, very inspiring. Thank you very much, Aoife. I don't know if you have any final remarks, but I think this is a very good way of ending, which basically we just went over the initiative, some concepts, then an example, for example, the Squirrel Project, all the ways people can get involved in this project, also how they can contact you and get their hands on these really cool and standardized. Most importantly, they look cool. Yes, that's great, but they're also very functional, and the whole workflow is now standardized, which means that you can collect all the samples and do all the analysis in the same way, which is very, very good for science. So do you have any final remarks about the project or about potential collaborators?
Just that if you're in any way interested or have the same sort of philosophy as we do, please reach out and get in touch, because we'd love to have you on board.
So you heard it here first. Do get in touch with Aoife, and then she will send you some kits and you can send your samples back and be part of this amazing initiative that is open source and very, very collaborative. So thank you so much, Aoife, for joining us. It was so nice to chat with you.
Thank you so much for having me. It's been great.
Thank you. You made this first podcast a very, very brilliant start. So hopefully now we have a very high bar to follow. And for those listening or watching, fear not. hologenomics, hologenome, evolution, and microbiome, and all these terms will be a constant in every single episode. But we will get researchers from different groups to talk about their different projects and all the very cool stuff that they are doing. So again, thank you so much, Aoife, and have a fantastic week. If you like this episode and are interested in following all the amazing work we do at the Center for Evolutionary Hologenomics, be sure to subscribe so that you never miss an episode. You can subscribe to our YouTube channel, Center for Evolutionary Hologenomics. The URL is currently a mouthful, so I'm not going to spell it out for you, but the good news is we can customize it once we hit 100 subscribers, so make sure you do subscribe. And there you can meet the researchers and watch some talks from work at the center. And you can also follow us on LinkedIn, Facebook, Twitter, and Instagram. We are out there. Bye for now, and until the next episode.